So the talk I'm going to present has this title, Demographic Inference in Structured Populations. The general idea here is really the work that has been done with several people. So Simon Boitard, Simona Gruzea, Olivier Mazet. Patricia has joined us in the last 2 years, and Willy Rodriguez. And this, most of them, except for Patricia and myself, are actually mathematicians or statisticians. And but the questions that we are asking are really questions that come from biologists. That is, questions which are about genetic data in populations. They don't have to be human populations. In fact, I'm not only interested in humans. But they're about the question, how much sense can we make from genetic data? And when we have genetic data from species that are distributed in space, to what extent do some assumptions that we make lead us to make conclusions that can be very wrong? And if that's the case, then what's the point of doing all that work? And there are many things, I think, that are really crucial because I believe that we really need to go back to basics and try to re-understand the genetic data in populations under structured models. Because structured models are very ancient. I mean, the oldest structured models are at least from the 1930s. So it's not like the idea that human populations or any populations are structured is new. The problem is what we do with it in terms of inference. So, again, the point here of putting all these names is also to say that the work I'm presenting here, of course, could have been presented pretty much by any of the co-authors, and it's really the result of many, many discussions. So it's a group result. It's something in which everybody contributed so much that I don't think any one of us can really say this is my work. Of course, my presentation will be biased by me, but so one notion that will come in these slides, and don't be afraid because of the complicated name, it really, I will try to explain the general idea. It's this thing called the IICR. IICR stands for Inverse Instantaneous Coalescence Rate, okay? I'll explain why I believe and why we believe that the IICR is a new way it both contains things that are known and it has also new things that allow us to look at genetic and genomic data in a way that may allow us to really do inference in very complex models. So, and I hope it provides insights into demographic inference and model choice, and also it allows to do something which is a summary of genomic information that's at least as informative as things that some of you have heard about, which are called allele frequency spectra. So my talk, because I realize there are many people in the room that may not be population geneticists, so at least they should think that they got something from the talk. So some parts are about more the biology or the general questions, and then I'll have some more technical parts at the end. So now the general question we have, and here it's a slide that sort of is influenced by human population genetics. It's really, we have genetic or genomic data. And the question is, how do we integrate data from other areas, like for instance linguistics, archaeology? For other species it could be geology, climatology, so there are many things. To look at patterns, that is, how is genetic diversity distributed in space, and how do we use all that to make a statement about, oh my God, I can't see that, evolutionary history, whether it's about population size changes, whether it's about population structure or selection. And really, this is what has been done in humans, in many species, and many models are used. Here I've represented 4 images of things that have been used either as verbal models, and others as mathematical models, and others are simulation models. So they're really different ways of looking at things. They make different assumptions. For instance, there are models that use the space. So this is work from Cura et al. In 2004. They use spatial simulations, and they compare the results of the spatial simulations to real data to choose among different models which one is the best. You have models that use summary statistics. So here, the authors used 4 different models. There is no need to go into details. I'm just here for principles. So there are different models. For each model, you compute a series of statistics that you can compute on your real data. And again, you compare observed and simulated. And then you say, the model A, for instance, is best to explain the data from population A. Here, it's a sort of intermediate model because it's spatial in the sense that you have a map, you have populations, which are the black dots, and you have lines that define where populations can go. So it's more complex than a non-spatial, but it's not as spatial as the first one I showed. You have models like that are not spatial at all, but they're structured, because you're going to say that you can represent human evolution by saying that, for instance, there is Africa, it came from a population that expanded. Here there is Europe, and there is something, people, I mean Neanderthals or something else, that had mixed. So here, for instance, they were using in that paper a particular type of statistics which are linkage disequilibrium patterns. So you see that there are many models. Some assume population structure, but no geography, for instance, split models. Others incorporate spatial processes, but they may assume a single non-structured starting point. For instance, many models about human evolution mention an origin from Africa, implicitly assume that Africa is a gigantic, is a big panmictic population from which all humans come from. So the question is there a way to find a consensus? How do we put all these things together? When you look through many studies and you're not a population geneticist, I think you can really feel lost. When you are a population geneticist, you do feel lost. And you know why. That's the only difference. Well, you think you know. And the question is, how do we do that also for other species? So when is it important to incorporate structure? So these questions, this is just to try to make sense of what I've been trying to do, is really to use these questions. How can we go from genetic data to say something about endangered species or humans? In humans, I mean, my work really owes to Guido. He was the person who introduced me to human population genetics and to the Neolithic transition. And in fact, the very first paper I published on these kind of things was with Guido and Giorgio. Giorgio may remember, that's the letter to America. You don't remember, and maybe you don't even care. So it was in '98, it's the paper in the American Journal of Human Genetics that we submitted about the question. It was already one of these questions, how do we interpret genetic data? At the time, the kind of criticism that we had with Giorgio and Guido was that people were interpreting trees from mtDNA, haplotypes, and they were talking about haplogroups, and they were interpreting the nodes of the trees or the groups of sequences as if they could directly be interpreted in terms of populations. And one of the main criticisms we had at the time, and we have been using for many years, is that these things are quite disconnected. You need to understand population genetics before you start interpreting this kind of data. And I think that now, I mean, this is a question that's always been with me, and I know that Guido and Giorgio were really important in all these discussions. Now, the work I've been doing in the last 12 years has been in Madagascar, and the kind of questions I've been interested are related to this question, which is you look at species that are endangered, so that would be the left side of my talk, of my slide on the research in the group. This shows about Madagascar. For those who have no clear idea of the size of Madagascar, I don't know if you know, it's the 4th biggest island in the world after Borneo, Papua New Guinea, and Greenland, exactly. It's about 600,000 square kilometers. So the comparison I usually have, which may not be good for Italians, is it's France plus Belgium. I know, it's biased. I don't know how big Italy is. So that's, okay, that's about double the size of Italy, okay? So that gives an idea of how big it is. It's amazingly diverse in terms of climate, it's amazingly diverse in terms of altitude, it's amazingly diverse in terms of biodiversity. What's been happening is habitat loss. I mean, and we know for sure, you can see it from maps, that all this was tropical forest, that's the eastern part, And you have very humid forests here. And we know that's pretty much for sure that forests have really declined. Now, there are other regions of Madagascar where we know that things have changed. I mean, this is a zoom in this area. And you see the red means that it used to be forested, and the orange as well, at different moments. So it shows how quickly forests were lost in that region. Now, there are all questions about how much forest there really was across all Madagascar, if you take out the east. And it's actually not clear. And what's interesting also with Madagascar is Madagascar was colonized by humans very, very recently. When I started working on Madagascar, people said about 2,000, 2,500 years ago. Now people would say more like 4,000, 5,000 years ago. They found new fossils while we were working on Madagascar. I mean, so it just shows still 5,000 is very, very recent. If you take Borneo, it's 40,000 years ago that people probably arrived, maybe even before. So it's not the same scale, but it's interesting. But it means that Madagascar provides an environment in which until 5,000 years ago, there were no humans. So if you can find important events in endangered species that happened before humans arrived, you have a good case to say that actually climate is a major driver. If you go to Borneo and find the same, it's difficult. You don't know if it was caused by humans or climate, for instance. Okay? Another thing that's interesting is Madagascar is very rich. There are many groups that are present across the whole island, so you can have comparative studies that are difficult actually to find in many other regions of the world. One of the things about Madagascar is the number of species that have disappeared. Again, so Madagascar, I showed this picture because it really shocked me when I saw it for the first time. It's in a classic book on lemurs. It shows actually all these species were primates living in Madagascar, probably still alive, for some of them at least, 1,000 years ago. They're huge. This one is estimated to have been the size of a gorilla. This one, you will see, is about the size of another kind of monkey that you can see here. So this is about the kind of sizes you have. Okay, that gives you, I'm not, that would be about 80 kilos. I'm not 80 kilos, but that gives you an idea. And this one is this one, that's the Indri. It's the biggest lemur presently alive. Okay, that gives you an idea of, he's quite small, oh my God, it's quite small here. And what is interesting is that Madagascar has probably today also the smallest primates ever, which are mouse lemurs. If you've seen the movie Madagascar, you recognize this guy. That's a mouse lemur. The genus is Microcebus. So there are various species of Microcebus, about 20, 23, depends on who you speak to, I guess. And they're the smallest primates. So imagine, that means Madagascar had until recently the smallest and largest primates of the world. That's amazing. And Madagascar is also characterized by the fact that it is estimated to have 100 species of primates. You can compare to Brazil, Indonesia, and of course in Madagascar they're all endemic. Okay. Now, it's just to give a bit the background of why Madagascar is potentially interested, and the kind of things we're interested in Madagascar is habitat fragmentation, habitat loss. So necessarily, nearly, this means you have to understand population structure one way or another. And you have to understand whether population structure influences your inference. Because it's not just population structure, it's changes in population structure. So here is a bit how people sometimes view fragmentation. You have a continuous habitat, it gets more and more fragmented. You arrive sometime later and you sample, for instance, here, and you sample here. So when you sample here, the typical thing that population geneticists have been doing, and I have been doing, and we've done that on orangutans, we've done that on elephants, we've done that on many species, which is you take genetic data in a region, you analyze it with methods that detect bottleneck or detect population expansion or quantify and date a change in size. So what it means is that implicitly you're saying when I sample here or here, it is as if the population, which is this fragment, was small in the recent past and it was big when it was more connected. That's the implicit assumption, okay? It may be explicit if you understand what you're doing, but that's what you're saying. So it means that you're saying that a history that is spatial is actually purely temporal and population size. So you're changing your, nearly, it's an epistemological problem. You're really changing your problem. It may make sense, but implicitly, it means that this is totally random mating. In that case, the results, of course, here and here should be similar. Sorry. Now, the reality is that actually your continuous habitat may be itself already structured, because even though if you take a big forest, I mean, not individuals do not necessarily mate with individuals which may be 200 kilometers away. So there may be some form of what we call isolation by distance, population structure, and so on. So the reality is that, and it's very complicated. So I'm not going to say we can answer that right now. That's not the point. The point is, first, let's go back and try to understand what population structure is. But it really asks, it's about questions like, how should we model populations? And in the group, we've been trying actually to address this kind of thing. So imagine this is your environment. This could be islands. This could be lakes if you work on fish. This could be many things. It could be hosts if you work on parasites. I mean, you could either assume that everything is like a big random population. You can use classical models of population genetics like island model, stepping stone, and represent your here. Or you can use also the idea that actually there is no such thing as a population. For instance, when you work on lemurs, on some lemurs I've been working on, like sifacas, I will show pictures, I think. So they live in small social units. I mean, the idea of a population makes no sense. When you know them in the field, there is no reason to think in terms of population. So maybe what you should do is that. And we've been developing work with social groups. We work on that and we work on that. So everything is fine. I mean, the question is always, what is my assumption? What is its limitation? How does it bias my work? And how robust are my results to that assumption? If you answer these questions, you're fine. It doesn't matter what you've assumed. So maybe I should just make a small stop. Is there something people want to ask in the middle? I don't know if it's the tradition here. No, I keep going. Yeah. So that gives a bit the background. I'm interested in genetic data. I'm interested because of structured populations, but also in humans, but not only. And I'll come back to that. Now, this picture is taken from a paper which I consider really a milestone in population genetics. I don't know, I know some people here know it, not everybody. It was published in 2011 by Lee and Durbin, and the general idea here was to use the genome of a single diploid individual and use patterns of heterozygosity along the genome. That means how long do you have to go with, you look at the genome and you have homozygous, 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 heterozygous, homozygous, 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 because most sites are homozygous in our genome. And the question is, how do you use this pattern to understand the demographic history of the population from which this individual comes from, okay? And the idea is that you have to make two types of assumptions, otherwise you can't answer. One is you have to assume that you know recombination rate, because this is what defines what is the length during which you can consider that the fragments have a common ancestor. That is, they have a common ancestor and there is no recombination. So you have to make an assumption on recombination. And the other one is an assumption on mutation rate, which says how often do you hit your DNA to change it. If you have these two assumptions, then you can do that. You reconstruct, it's very complex. I don't understand it all. My colleague mathematicians do. I have problems with that. But the point is that this is just one single individual. You apply the method and you have this kind of wavy curve. So what do they represent? This is time. This is close to the present. This is very far in the past. I don't know if you see, this is 10 power 6, so a million years. And here this is 10 power 4, 10,000 years. Okay? These are humans. This means Yoruba, European, Korean, Chinese, okay? Yes, so that would be Yoruba is African. So you see that they all show a similar history here, and then here some curves seem to depart, and they all go down, and then and then they seem to go up, or where am I? Here. And here they go up in every direction. So it's difficult to know if it's a statistical noise or a real population size increase. So you understood, of course, this is this is population size, and this means 10 power, no, that's 4 times 10 power 4. So, okay, so that would be 10,000, 20,000, 30,000, 40,000, okay? So it shows it, if you believe that, you would say, a long, long time ago, there were 20,000 human beings, or Homo, whatever it was, about a few million years ago. Then the populations went down, and then increased again about 800,000 years ago, and then they increased up to about 200,000 years, and then they went down, but somehow Africans went down slower, and this one went down much more, and it went up. And the kind of interpretation that people have is like, this decrease here would be the out of Africa, which of course is problematic because Yoruba were still in Africa, so maybe they went out and came back because they forgot the keys or they forgot to close gas. The other thing is that people say, look, there is a split. So if we look at here, that would be the history that is shared by all humans. And so people say, look, that's funny, it's about 100,000, 200,000 years. That's about the out of Africa, so that should be working. So the split of these curves is interpreted as a split of populations. And the decrease is interpreted in one case, and in the other case is not even discussed. I mean, if you go back to the paper. So I personally had big problems understanding these numbers, because I was looking at them and I said, is there really any reason to think that there were many more people here between 100 and 200,000 years than, let's say, 500,000 years ago? And more than during all that period? And I personally couldn't believe it. And the first thing I thought is population structure, because we had been working, and I will not show that, on the question of how does population structure influence the history, demographic inference. And we had shown, I mean, the major work for me is Wakely, 1999. So there is a major work by John Wakely. And I was informed by Mark Beaumont about that work, and we discussed it a lot. And I started understanding that when populations are structured, and I'll explain why, you get bottleneck signals. And it's structural. It's not something you can avoid. I mean, you can play with it, but it's difficult to avoid. So this method has been used on many species. This is a paper published in Nature 2, and here it's again the humans. This is the different chimpanzees. This is gorillas, and this is orangutans. So for instance, the orangutans, you have 2 subspecies, and you can see that they have very different histories, and here they join, and it could suggest that here is when the 2 species separated. Okay, that would be the kind of interpretation that people have. This is more recent on the gorilla. Okay, you get a bit that, but it's more precise. Now, to understand these kind of methods, there are various things that are crucial. One is to understand that these methods use something called the coalescent theory. So the coalescent theory is a theory about gene trees. Okay, I don't know how. Are there people who have never heard the word coalescent? You can. I mean, you won't be thrown out of the room, I promise.
The answer is yes.
Guess what? There are people who have never. Okay. Okay. The idea is that the coalescing is just an English word that pre-existed coalescent theory. So it's just meaning that you take genes. Okay. When I say genes, it's important. It means it's the same homologous region. Like you take, for instance, my mtDNA and Guido's mtDNA. And the question is, when do they have a common ancestor? When is the molecule that mutated from there and descended through different people and ended up in Guido, and then the other ended up in me? And the point where they have a common ancestor is the coalescence event. They coalesce, the lineages join. Okay? You can see it the other way. It's a splitting of lineages. Okay? So here, the coalescent theory, I mean, one of the persons, in fact, that knew much more and still probably does is Giorgio. Giorgio, when we met, it was in Montpellier, when he came to work with Monty Slatkin. while he was doing his PhD with Guidon, and was among the first people to really work on these questions of coalescent theory. And he looked at the importance, for instance, of different statistics to reconstruct the history of populations, or I have to remember exactly the work you had done with Monty. It was a long time ago, 95 or something like that, right? Yes. Huh? You'll remind me, I have to reprocess all that. So the point is that, imagine that you have these things. So these things, these lineages are, let's think, empty mitochondrial DNA. So it's really, so the mitochondrial DNA does not recombine. So it means that if you take a molecule here, they must have a tree that explains their history. The coalescent theory starts with a standard model. Population size is constant. It's a constant. And in fact, the theory shows various things. One is that the height of your tree is a function of the size of the population, which is here represented by the width. So this is a population, this is the past, this is present, time goes this way. So you sample here, and this is the tree of the genes. It's not a population. Here, there is only one population, nothing else exists in the universe. That's the only population of that species, okay? So, and there are a number of things, is that you have to introduce the coalescence rate, which means that the probability or the rate at which, at every generation, for instance, you can have or not a coalescence, so a common ancestor. If you can see that if a population is small, you should have your ancestors very quickly. So the coalescence rate is high. So there is an inverse relationship between coalescence rate and n, the size, okay? This is the function, but you can ignore it. It's just that it says that it's a function of your size, the size of your sample. K is the number of lineages. For instance, if you have a sample size here, k equals 5. So the function, this is the rate when you have 5. Once you have the first coalescence, now you have 4, and so the rate changes, okay? So the coalescence rate is an inverse function of the size, okay? And it's a positive function of the sample size. Now, people in the 1990s showed that the trees, and so the coalescence rates, are highly function, as I just said, of the population size. So you expect that the gene trees will have this kind of shape when population was big in the past and small in the recent past, and this kind of shape in the contrary when it's small. Why? Because when the population is small, the coalescence rate is high. So you have a lot of the coalescences. Now, if these two lineages arrive here and have not yet coalesced, now their coalescence rates depends on these populations, so they will take a long time to have a common ancestor. Now, that's the opposite here. I don't know if it's clear. So, you see that the demography, when it is seen as population size change, influences the shape of your tree. It influences the rates of coalescences, okay? And it will influence patterns of polymorphism. And this has been at the heart of research since the 1990s until recently, and in fact, until today. Looking at polymorphism helps us understand changes in population size. Now, very early, and maybe to make it clear again with the terminology, imagine the population was big in the past and small here. As I said, the coalescence rate is a function of the size, n0, and in fact it's a function of the inverse. Remember, it's inversely proportional. So a small n0 means high coalescence rate. And here, when the population is high, it's the opposite. Coalescence rate is a function, so large n, low coalescence rate, so very long branches. And here, this means that if we can estimate variation in coalescence rate, we can then estimate variation in n, because it's inversely related. And there are many methods. There is something called Bayesian skyline plots or skyline plots. There is PSMC that I mentioned. There is MSMC which is for multiple genomes. It's like PSMC. The one I showed before, we know with the curves applied to humans. And there is recently something called stairway plots that you can use on many independent fragments of DNA. Now, very early, people realized that when populations are structured, if you sample here, for instance, this population does not live alone. Remember, before I explained, the population is completely isolated. It's the whole species. There is nothing else. In fact, that's rarely the case. So this population, when you sample genes here, you see that actually they coalesce at a rate which we know is this one. But maybe this ancestor actually came from here. So now, for this to coalesce with that, they are in different populations. So they cannot coalesce now. They will only coalesce if another migration events bring them back together. So it means that, in fact, the coalescence rate from now to the past will depend on the meta-population of the species. And that explains. So it means it's a bit as if the population was small here, and then in the past it's big. So that explains when you sample in one population genetic data, you get a bottleneck, just because of population structure. Because from the gene point of view, the coalescence rate is high, in the recent past, and it's very large in the ancient past because of migration and connectivity. So it means that it's unavoidable, it's a property of gene trees that in structured populations, you get bottlenecks. This leads many questions, because if you say that there is a bottleneck or a population size signal, it means that a structured population, from the coalescent point of view, has a minimum of two sizes. It cannot have one size. So it means that the notion of effective size is very problematic. You can't have an effective size for a structured population, because by definition, if you had an effective size for a structured population, that means that you could construct a population constant that would have the same tree. But we just saw that the constant, there wouldn't be a constant size population. So that really questions the notion of effective size. Okay. And so now we enter the work the work that we've done, so with all these people, and now recently Didier Pinchon joined us on some issues that are more mathematical, and I think there is somebody here you may know. I'll let it a few seconds so that... So this is what I'm presenting now is based on these two studies. So one published last year, one to come out now. So, okay. So the idea of the IICR. So we've understood, so populations are structured. When you sample genes in one of the populations, you will have a genealogy that will depend on the structure. And of course, it will depend even worse when the structure changes. So let's try to start again. Reality is complicated, so maybe reality could be that. Okay, it was continuous, becoming fragmented, and you take two genes here. You take two genes, okay? Two genes again is like two empty DNA, for instance. Or you take one diploid individual and you start at the beginning of the genome and you look at two bits that are non-recombining. Yes. So you could look, imagine that you had magic powers, you know when the ancestor is. So for the first one, maybe the ancestor is last generation. Okay, you write on the table 1, so one generation. You keep to the next bit and you find 1,000 generations. Okay, the ancestor was 1,000. And you keep going along the genome and you will have values of when the different bits have an ancestor. So you can build a histogram. Okay, you have a histogram of coalescence times for your genome. That's the distribution of coalescence times for your genome. Okay? These coalescence times, so you take the model, any complicated, imagine that the population may have been increasing, decreasing, you just take that one diploid individual, and there is necessarily a distribution of coalescence time. Maybe you have problems estimating, but it is there, it exists. That's the reality, that's what happened. This distribution, as you saw before, remember I explained before that when population size changes, you change coalescence rates. If you change coalescence rate, you change distribution of coalescence times. Okay? The rate, if you have a particular size, constant, you will have a distribution of coalescence times. Maybe you'll have many recent coalescences and a few ancient. If you change the size, you'll have many ancient and very few recent. You see, these are distribution histograms. Now, in this very, very complicated model, incredibly complicated, you can't even understand what it was. There necessarily is a distribution. You don't know what it is, but there must be one. Now, imagine that you can estimate it. It means that you can fit a history of population size changes that will explain it perfectly. This is what I'm trying to say here. So then you take a sample of size 2 in one of the DIBs. There must be a distribution of coalescence times associated to that model and to that sample. That's a crucial, crucial word, sample. There must be, thus, if there is a distribution of coalescence time, there must be a distribution of coalescence rates. And I explained before that if there is a distribution of coalescence rate, that is, if there is a coalescence rate, there is an effective size that is associated to that. So there must be a fictitious history of population size change that will perfectly fit the distribution. This fictitious history is the IICR of that sample, and I call it IICR2. I stop a second. So it means that whatever the history of a species, if I take one diploid, remember just one, I can find a model of population size change that will be as good as the real history. Because we never know the real history, in fact, the population size change model will probably always be better than any model that you and I can imagine. For instance, when you look literature, when people try to estimate, to infer human history, they'll say, okay, you remember the figures I showed? They'll say, oh, there was one population and it's split in 2, or in 5, or in 6, or in whatever. That is never going to be as complicated as reality, ever. So that model will always lose in front of the population size change model. And because the IICR is estimated by the PSMC, it means that the PSMC will always beat everybody. So if you wanted to come up with a model, I would bet that the PSMC will at least be as good as your model to explain the distribution of coalescence times. And this result, I mean, I was amazed when it was an intuition by Olivier. I remember in July 2015, we were discussing these questions, and he suddenly said, but we're losing our time. We can't beat that model. Population size change will always beat every model. That's necessarily the best. You may think, what the hell is he talking about? So I'm showing this just to scare you a bit. This comes from the coalescent theory. Don't be worried. It's another way of reformulating what I said. So it means, in population, so this means what is the probability that a particular coalescence time is bigger than a particular value of time. Okay? That is, you take your genome, for instance, you could say, how many of the coalescence times in my genomes are older than 1,000 generations? That's a number, okay? You may not know it, but that's a number, and that's a way of representing your distribution. People in the 1990s have shown, mathematically, that you can express it in terms of a population size that changes size, okay? A population that changes size, and this is this lambda that you don't see very well, but this is a classical result from 1995, I think, okay, by Donnelly and Tavares. What it means is that there is a simple relationship. I mean, simple, it's not simple. I mean, let's be clear, it's complicated for me, don't worry. It's just, so this expression shows that there is a mathematical relationship between these, which we can understand what it means, and this, which is difficult to understand. Now, if you play with this math, you can remember that there is something which is 1 minus that, so that's 1 minus that, is known to be the cumulative distribution function, which we all have seen in statistics, okay? I don't go into details. When you derive the cumulative, you get the density. Okay, so the density is 1 minus that, is the derivative of that. If it's the derivative, you just do the derivative, you get this complicated function. Forget about it. Then if you then transform, you put lambda, you move things, you end up with lambda is that. Okay? Believe me, you don't need to go into details. The point is that this perfect history of population size change is very easy to compute. You just need to have the distribution of coalescence times. If you have it, you compute that ratio. So you simulate under the model of interest, you compute this statistic, which is the IICR, and you just plot it. And there it is. There it is, as I said. So here is a case of an island model, okay? I know it seems a bit complicated, and I try to bring it back again to reality, which is, if I take a particular individual, let's imagine that the human species is a set of island models. It's a set of islands connected through gene flow, like Wright's island model. So there are n islands, n could be could be 2, 3, 4, n, bigger than 2. So if you take an individual, the two genes in the same island. So imagine you go in an island, you take a diploid, and you do the PSMC, more or less. You know, the method that I mentioned. And imagine that that species really, really works like an n-island model. What you would get is an IICR, which Olivier derived mathematically, but you can get by simulation. And he showed that the IICR looks like that. Okay? And this line I've drawn here is constant size of the n-island, and it's represented here too. It means your island model is completely constant. The islands have the same size all the time. They exchange the same gene flow all the time. There is no change in size. They're connected. It's forever like that. But if you sample an individual in one dim, this is exactly the history that you will believe. That is a history of population size change that will produce exactly the same data as the island model. Okay? You understand? So it means that if you sampled an individual, imagine like humans were from an island model, you would say, oh, it's amazing, humans had a bottleneck, there were many more humans let's say here the scale is bad because it's in generations. So you would have to multiply these numbers by 25 years per generation. But let's say, so you would say, oh, about, I don't know, 500,000 years ago, there were many, many humans, and then they went through a bottleneck, and now there are very few of them. And because the PSMC is a method that uses molecular data, so it makes errors, when you arrive actually to recent past, it starts to do weird things here, not always. So you would think, oh, there was a recent expansion, that must be the Neolithic expansion, surely. So you see that you can end up with stories that have nothing to do with any change in size. They are just because of the fundamental properties of the coalescent in a structured model. Now imagine that you sample an individual now who happens to have his mother or his father that comes from another island. It's first generation hybrid. Half of his genes are local, half of his genes are from another island, right? So it means that actually they cannot coalesce because they were from different islands. And they can only coalesce when they meet again in an island. And what do you get? You get what you get here. So again, the line here, this is the same model. So the size is constant. But you get a population expansion because here, the coalescence is impossible, so the apparent effective size is infinite. You could think, oh, that's bullshit, it wouldn't work with real data. Real data are not that bad, they're not that stupid. You apply a method that we did it with PSMC, we simulated gene zones exactly like that, and that's what you get here. The green is the PSMC estimation, so it means we really simulated genomic data. We gave them to the PSMC. We used the analytical theory, which is the nice line, and then we did an estimation using the estimated IICR, using a limited number of data, and that's the red line, okay? And in fact, what happens in an n-island, so here it's, when you change gene flow, you change the bottleneck. You change the size of the bottleneck, so when there is little gene flow, The signal you have is of a very ancient bottleneck from a very large population. If you increase gene flow, the bottleneck decreases and becomes more recent. This is what you see here, okay? So migration 01, 02, 03, migration 20, 50, whatever. And of course it's normal, because when migration increases, your populations behave more and more like a random mating group, a random mating, right? If you migrate among all islands, it's nearly like migration. So what you get is the size becomes equal to the number of islands. Because here the size, is scaled by the population size, okay? When you have 50 here, it's because there are 50 islands, okay? Each island counts one. You could say it's 50,000 if an island has a size 1,000. Okay, and when you sample from different islands, you also get the changes of these expansions. So now what happens when, again, the island model is of constant size, which is this line again, but you change gene flow 3 times? What you get is, these are the moments at which we change gene flow, and you get humps. You can start recognizing a lot of the PSMC plots that people find. So what we're saying here is that, in fact, if a set of islands was connected, disconnected, connected, you get PSMC plots similar to what you find in humans, or in gorillas, or in whatever you want. So, to explain PSMC plots, which have been interpreted in terms of population size change, in fact, you don't need that. It may be true that humans changed in size, but what you need more here is connectivity. Or, in fact, look at that. Here is a model in which the islands were here, and suddenly, at that moment, they all double in size. They all double in size, okay? So this is where the line goes up. But in fact, the PSMC and the IICR are here. They still see a bottleneck. So you see, population size constant, you change gene flow, you have these humps. Population size increases, you still see the decrease. Which shows again that in that case, structure is so profound, so important in the way it influences your genes and the genealogy of your genes, that you still see just the bottleneck. The population size increase is nearly invisible. Okay. And then we applied it, of course, to humans. And what we found is that we could reproduce human PSMC plots by having 3 changes. One, so the blue vertical lines correspond to major geological events or major paleontological data. So the first one, the first blue, is the beginning of the Pleistocene at 2.5 million years ago. 2.5 million years. And you can see that the first change in migration that we found is exactly there. Then we found that you need a change in migration around 90,000 years ago. 950,000, nearly a million, slightly less than a million years ago. And you see that there is this blue line here, which is the mid-Pleistocene. So it's not very far, it's not perfect fit. Remember that it's an N-island model. So and this one is just the oldest Homo sapiens fossils known. They're about the time at which we find that you need to change gene flow, okay? So, what does that mean? It means that it's, in fact, not that complicated to reproduce the human PSMC with relatively stationary populations, but with changes in connectivity. Now, the models that we've been looking at are N-island models. So they're very simplistic, because in an N-island model, there is no space. There is structure, populations are structured, but there is no space, because everybody is connected to everybody exactly the same way. So it's as if you were just as distant from anybody. So we did some work on the stepping stone. So for those who don't remember, so here is a 3x3 stepping stone, and here is a 7x7 stepping stone. Okay, so you can either sample in the corner, in the center, or on the edges, and this is what we did in this 7x7. And so the dims are counted like that: 1, 2, 3, 7, 8, 14, etc. 25 is center, 4 is this edge, and 1 is a corner. And when we do the IICR for these dims, this is what we get. So the center one is here, and the corner is here. So you can see that the individuals, depending on their position, will have IICR, and therefore PSMC plots, that will differ here. And they join here, but there is nothing, no meaning whatsoever. I mean, there is a coalescent meaning to that joining, but there is no meaning in terms of split, it's not like, oh, this is the moment in which they split, and here there was a separation between the populations. So it's a bit like when people were interpreting haplogroups to say stories, interpreting PSMC to say stories that way is just similar. You're making a statement about something that has nothing to do with what you're talking about. And the IICR have properties. And when you sample an individual from different DIMs, so for instance here it's the green one is if you sample from DIM 1 and DIM 2, in fact this, 1 and 2, then you have 1 and 3, 1 and 4, 1 and 25. And you get these curves, and you see that it's interesting because the green one, for instance, you get a stationary ancient population that decreases and then has an expansion. Okay? And when you get the red, it's when you take, it's as if you were saying, I'm making that up, but imagine somebody who would be the son, let's say, of a Breton and a Russian, or maybe an Indian, I don't know what would be the equivalent in humans. In fact, you could expect, based on a simple stepping stone model, you would expect this kind of decrease-increase. So if there was particularly recent, for instance, migration, that could happen. Okay. Now we looked also at split models. If you have a split model here, this is the IICR. So here, the split model green means you have an ancient populations of size n, and at some point it splits into two populations of size n. Or you have an ancient population of size 2n, and it splits in two populations of size n at time t equals 2. And in fact, what you get is like, at the beginning, and here there is gene flow, sorry. There is gene flow between the DIMs, okay? And what you get is the IICR will go up and then down and then stabilize at the size 2n for this and n for that. But you can see that the split moment does not lead, because the curves are true for all DIMs. So the splitting moment, again, is not connected to a split in the IICR because there is no reason why it should do that. Has nothing to do, again, it's a mixture in the same way that people were mixing trees, genes of trees, to make to talk about populations in the 1990s and 2000s. It's the same kind of mixture. It's like you take something that has nothing to do with it and you start making up stories. And when you have a double split, you can get this kind of multiple curves, okay? I'll go quicker. And now, of course, you could say that's all very theoretical, so how do you use it? And the general idea is to say the IICR can be used as a summary statistic of your genome because it informs you in a rather interesting way, which is sample dependent, because for instance, in stepping stone, depending on who you sample, you get a different history, a slightly different history. So maybe we can use it. So here we took an example of something already published on admixture with Neanderthals. And I don't know how many people are familiar with that, so you know that we are told that there was admixture with Neanderthals. And there are a number of arguments that have been put forward, okay? Again, my feeling when I read the arguments is that they're problematic in many ways. I mean, I don't think any of them is really strong. The only strong argument is actually one data set from OASE, where they find a high proportion of Neanderthals in one individual, and there it's difficult to negate the possibility that something happened a few generations before, but it was one individual, and so on. So what happened there, apart from that, I have really difficulty. A lot of the arguments from population genetics, that's the only part I can criticize or not, are incredibly poor, actually. They were first based on one statistic called the D statistic, and supposedly the value of the D statistics was a good way to find admixture. Problem is that very quickly people found that actually models in which there is no admixture provide exactly the same values of D. So what came later is this paper, which was also an improvement. What they said is that we're going to look at something called the allele frequency spectrum, but they modified it. They use, it's called the double conditioned frequency spectrum, I think. So it's another way. It doesn't matter. It's a clever way of looking at things. It's a good work. I mean, there is no. They just said, okay, the D statistic is not good to separate admixture from structure. Let's try something else. So they simulated data of humans, Neanderthals. So here you have, Africans, non-Africans, and you have Neanderthals here, and there is this model with admixture. And here you have a model in which there is something happening there, an ancient structure. I mean, I really had problems understanding the model. But anyway, it's a model. And what they did, not understanding what it does, understanding where it came from and why it would be reasonable to think it's a way to represent that. And they looked at that, and so you see here on the left, okay, just on the left, you have models with recent admixture, and on the right, models with ancient structure. The point is, and I don't know if you see it well, there is the real data, and then there is the simulated data, okay? So they use many models. I mean, they really did a lot of work here, changing a lot of parameters, putting bottlenecks, no bottlenecks, whatever. Okay, many. And what they find is that when there is recent admixture here, you see that the black curves look like the gray curve, and the gray curve is the real data. Okay? But when they look at ancient structure as defined by this model, they find that, you see the lines here, the black lines are flat. They don't have this gray point here. And so they said, well, the best model is recent admixture. And that's very true. I mean, based on that statistic, that was an improvement because they could show that recent admixture here was better to explain some humans' polymorphism than ancient structure as defined here. Okay? But again, it's ancient structure as defined here, if you believe that model makes sense. Now, what we just did to show again the power of the IICR was to simulate under the models that they had suggested and compute the IICR. Very simple. And we also did the PSMC. And you find that all the models they suggest are here. The real data are here. This is the Neanderthal. This is human. And these two curves, which are slightly different here, are the model of Lee and Durbin and the model that we had proposed before. I won't enter into details of why there is a small difference here. It's really, we can just fit that one as well. But Lee and Durbin proposed one model of population size change, and so we fitted Lee and Durbin's model. But in fact, the model that Lee and Durbin used doesn't perfectly fit all humans. But it's another issue. But the point here is, again, the work of Yang et al. Was really good. I mean, there is nothing wrong about it. I mean, it just chose models which may not be the best models to represent human evolution, but that's what they did. So what should I conclude now? Well, the thing is that we all are entering a world of genomic data, loads of stories to tell. The question is really, how do we really use this genomic data in a meaningful way? I believe that instead of validating models, we should try first to exclude. We should try to show that models just cannot explain data, and then see which ones are best. And the IICR is a very good way to do that. The PSMC estimates the IICR, so we do have a model. It's there. Just use it. You just have to understand that it's an IICR for a particular sampling scheme. The MSMC is the same as the IICR, except that it's for K genomes. And I believe the IICR curves can be seen as summaries of your genomic data, and in fact, they're a bit summaries of your model conditional on your sampling. So we can compare the observed PSMC using real genomes to IICR expected under a set of competing models. That's what we did, for instance, for humans, for Neanderthals. We can use this IICR to exclude models and identify likely ones. So we did that for humans and Neanderthals. And I think that will be most of it. And just wanted to note, because sometimes people think the IICR, if you remember I said, the IICR is the inverse instantaneous coalescence rate. And I told you that coalescence rate is inversely related to population size. So inverse instantaneous coalescence rate should be the equivalent of instantaneous population size or effective population size. But I insisted on the fact that no, it is not. And I explained again that a structured model from the coalescent point of view cannot be equated to an effective size. So a non-structured population. So there is no such thing as an effective size from the coalescent point of view. Thank you.